Hello, and thanks for joining us today for this iRepertoire webinar. My name is Matthias Jauregui, and I will be your moderator today. In this webinar, Dr. Martin Davey, Associate Professor at Warwick Medical School, University of Warwick, will present data on their study of unconventional T cell immunity in malaria. In particular, the role that gamma delta T cells play in the human immune response to malaria. So without further delay, I will turn the time over to Dr. Martin Davey. Thank you very much.
Good morning, good afternoon, good evening, everyone. Hopefully everyone can hear me well. So I'd just like to firstly introduce myself, and my name is Dr. Martin Davey, and as Matthias mentioned, I'm an associate professor at Warwick Medical School. And some of the work I'm going to talk about today was done when I established my lab at Monash University several years ago, and have now transitioned to Warwick. So firstly, I'd like to thank iRepertoire for the opportunity to present some of our work here at this webinar and sort of introduce some of the analyses we've done, and particularly around some of the techniques we've used, which have included some of iRepertoire's proprietary TCR repertoire sequencing. We've got a few little nice images of Warwick and our biomedical research facility, which is a very nice place to work and somewhere where we're setting up lots of our sort of follow-on studies on this. So today I'm going to talk to you about our work around unconventional immunity, and particularly gamma delta T cell immunity in malaria. So I just want to sort of go quite broad here and let's get this laser pointer working, and just sort of highlight that, one of, medical science and humans' greatest sort of achievements is actually probably vaccination. And you can see from these graphs the sort of impact on human health that the introduction of vaccination strategies towards various quite prevalent and highly effective pathogens shown here. And you can see the points where the arrows and vaccination strategies are introduced. You can see the almost complete eradication of some highly pathogenic diseases that have sort of blighted human existence. And since then, our sort of understanding of the role of the immune system, that some of these sort of rudimentary, in essence, sometimes heat-killed whole pathogens elicit, and how they drive the immune response. But I would say, as we've sort of gone on, our real understanding of how these vaccines work and immunity works is probably still not really well understood, and certain pathogens have sort of evaded our attempts to generate vaccines, one of those being malaria. There's one licensed vaccine which is less than 30% effective, yet still that's going to impact human health and save millions of lives. But having a vaccine that's greater than 50%, or even up into the sort of 80s and 90s, like we saw with COVID vaccinations, having that kind of protection level would be vital for malaria, and particularly other things like tuberculosis that have evaded our attempts to design vaccines. And some of the theories around why that is, one or two of them have sort of centered on our understanding of what generates immune protection and arms of the immune system that actually are elicited in some of these infections. And so that sort of builds on some of our basic discovery research, looking at maybe some poorly understood arms of the immune system. And so that sort of brings me to sort of gamma deltas and the immune system. And, we all know that the immune system is a highly complex system full of cells and soluble mediators. And so conventionally, the immune system is sort of broken down into two rudimentary arms. So I would, the innate immune system, which is sort of the hallmark of innate immunity, is the possession of innate receptors, so germline encoded invariant receptors that recognize pathogen-associated molecules. And this allows that arm of the immune system to really rapidly respond to invading pathogens. And then through evolution, we've developed a system where we can generate what's called adaptive immunity, but it's essentially an immunity that can change in the face of a new threat or a new pathogen, and evolve in a way that sort of adapts to that pathogen and allows us to generate a memory response, which is incredible. But where do gamma deltas really sit within this sort of system? Well, it's long been thought that they sit at the interface because they utilize specific receptors in a sort of innate kind of way. And the receptors I'm talking about are an adaptive receptor. And so gamma deltas and their alpha beta T cell cousins express a very specific receptor, and alpha betas express an alpha and a beta chain which forms their T cell receptor, whereas gamma deltas express a gamma and a delta chain to form their T cell receptor. So slightly different, but a lot of the principles are the same. So alpha beta and gamma delta receptors have these highly variable regions called CDR3 regions, which allow them to recognize a whole magnitude of different antigens, which is an antigen is essentially a molecule that would be eliciting a T cell receptor response, and is sometimes pathogen derived, or it can be also host derived, such as through cancer immune surveillance, etc. So gamma deltas really sit at this interface. So they use this T cell receptor in an innate kind of way. And that's been the central sort of theme of gamma delta research for many years. And, where do gamma deltas and why are we interested in them? Well, there's been lots of sort of small studies over the years and some larger ones that have sort of identified that gamma deltas expand rapidly and en masse in certain infections, such as malaria, tuberculosis, and cytomegalovirus. They seem to work in an MHC independent way. So alpha beta T cell receptors are exquisitely constrained by their interactions with MHC molecules, whereas gamma deltas seem to operate completely independently of that system, which is quite remarkable. And also there's been an explosion recently of immunotherapeutic companies that are trying to harness gamma deltas based on much of our sort of research over the last 25 years. So I just wanted to give you a little bit of a background on where these gamma deltas are. And so most of our knowledge has come from mice, and obviously we can dissect and understand a lot about gamma deltas from mice. And so one of the most remarkable things is that during embryonic development, we get specific populations of gamma deltas associating to very unique tissue locations. And actually, it's populations that are expressing very specific gamma delta T cell receptors that end up in these locations. And so you get these waves of selection and propagation in different tissues, which is incredibly interesting and suggests sort of host encoded antigens and potentially innate mechanisms. And this is paralleled to some extent in humans. So there's a population of gamma deltas that are very frequent in the blood. So about 5 to 10% of all your blood T cells. And again, they've got a restricted T cell receptor usage. They express a V delta 2 and a V gamma 9 T cell receptor chain pairing to allow them to form their T cell receptor. And they're less common in the tissues, but in the tissues it's less defined. So what we end up having is this sort of broad array of V delta 2 negative, as we call them, crude population of non V delta 2 gamma delta cells, and then probably the most common chain pairing is this V delta 1. But then there's a whole array as 8 different gamma chains that this V delta 1 compare to, and there doesn't seem to be the same kind of gamma chain restriction that you see in mice, and actually it's more that they're expressing a V delta 1 with a vast array of different receptors, and I'll show a little bit more data to sort of support that. And it's not really clear what these V delta 2 negative populations, particularly V delta 1s, are doing. So there has been some understanding around sort of viral immunity, that maybe these cells are expanding and helping to recognize viral or host antigens that are upregulated in viral infection. But this is sort of vastly understudied, and I'll sort of push a bit more into that now. So one of the key questions actually in the field is sort of gamma delta T cell receptor antigens. So as we I sort of alluded to earlier, alpha beta T cells are incredibly well understood, and that's simply because the mechanisms and the engagement of antigens is very well described, and we can harness that to understand their responses in infection. So you've got MHCs presenting peptides. On the other hand, there are populations of innate receptors and cell types that are expressing alpha betas and use constrained receptor pairings, such as mate cells, which recognize riboflavins in the context of MR1, and iNKT cells. Again, they use a very restricted chain pairing to recognize CD1 molecules and lipids. So, as I mentioned before, the V delta 2s, potentially are an innate-like population and recognize in the context of these butyrophilins microbially derived HMBPP, which is a riboflavin, not a riboflavin, a non-mevalonate pathway molecule. So it's a tiny phosphate containing phosphoantigen is what we call it, but it's just an incredibly small molecule, and it's been shown that it binds one of the intracellular domains of these butyrophilins, which allows it to interact with gamma deltas, but the sort of mechanisms of that are still vastly unclear. But then when it comes to the sort of V delta 2 negatives, we're not really too clear what drives their biology, whether it's very distinct sets of antigens or highly diverse ones. But there has been a handful of antigens described to date, and I've been involved with this description of MR1, which is obviously the key molecule that presents riboflavins to mate cells, so in a very conventional top-down fashion. But actually what we found was that these gamma delta cells were recognizing MR1 in a highly different way to, say, a mate cell interaction with MR1. And actually some of these receptors were binding underneath the MR1 molecule, which is incredibly different and more similar to sort of B cell and antibody responses. So maybe that gives us a clue as to what they're actually seeing, which might be a highly diverse pool of antigens. But where does that happen, and what that looks like is almost completely unclear. So one of the ways that we sort of set about doing things in the past and over the years is to sort of look at their T cell receptor repertoires and try and understand, are there populations of cells that are sort of restricted? Are they using specific receptors? And can you see expansions of clonotypes that would indicate, specific antigens being used? And so just to sort of plug a bit of iRepertoire's, receptor pathways and some of the data analysis you can do. This is a tree plot which shows a gamma delta repertoire, and you can see each of these quadrants of color. Each of these quadrants represents a single gamma delta CDR3. So this would be a gamma CDR3. And the size of the quadrant of color indicates how much of the repertoire that CDR3 receptor is taking up within the repertoire. And you can see actually there's quite significant expanded single clonotypes within this example repertoire. So this is actually a gamma delta taken from our research. So, and I'm just going to talk a bit more about how we've looked at these repertoires and used these tree maps to visualize the behavior of gamma delta T cell receptor repertoires. So one of the things we did and what we looked at was sort of this sort of subdivision of gamma delta. So we looked at these V delta 2s, but also the V delta 1s, and split them apart. And we looked sort of at two sort of distinct or distal time points in human development, one being neonatal. So this would be from cord blood. So, one of the first points of neonatal life and into sort of adulthood. And also in sort of adults. So this would have been between 20 and 30 years old. So not really old, but, very established into adulthood. And one of the key things that we found within these repertoires when we started looking at them was actually that the V gamma 9 V delta 2 cells didn't really change a lot between a neonate and an adult, and had over 80% of the shared sequences between separate individuals. And we've done this on quite a scale now, and it's holding quite true. And what you find is that when you look at one of the gamma repertoires, for example, you sometimes find this sort of almost, it is germline encoded receptor. So not germline coded, but a germline sequence. So it has no sort of changes within it, which is the changes are a hallmark of CDR3 sequences. But actually, when you look at the paired repertoire, these gamma sequences are pairing with multiple different deltas. So actually, these repertoires are highly diverse. But also the sequences are very shared between individuals, which is quite remarkable. But it does sort of suggest and support the idea that they're recognizing very similar things across multiple different cells within each individual. But then when we started looking at the V delta 1 cells, we found something much different. And what we found in the neonates of these repertoires were highly diverse. But by adulthood, what you were finding was like major clones hyper expanding within individuals, but also individuals that had remained diverse. So it suggested that, potentially there's triggers or genetic differences that might be actually driving some of these populations of receptors, or single or oligoclonal receptors. And when we sort of started looking across individuals, actually a lot of these oligoclonal receptors were completely private. So you didn't find them in anyone else, essentially. And this has also been replicated by others as well. So to sort of combine that, we did a lot of sort of flow cytometry and other sort of functional assays to try and understand this biology, now that we knew that there was sort of clonal populations and differences. And one of the things we sort of drilled down on was that actually sort of these diverse populations of gamma deltas. So if these individuals with these kind of repertoires actually had very naive like cells. So you can see in the blue radar plot here, they were expressing classic markers of sort of naive T cells, so CD28, IL-7, well, it's been chopped off, IL-7 receptor alpha, CD62L, and CCR7. And I'll sort of discuss what that means for in an immune cell population. But then these sort of clonal populations were actually shifting towards almost opposite kind of phenotype. So they'd lost all of these markers and upregulated highly cytolytic granule markers, whereas the innate-like cells actually bridged pretty much all of it. So they had the potential capacity to do all sorts of things. And we also looked at sort of the transcriptional profile of these naive and effector cells, and they were highly distinct. So you can see the effectors on this line here, so expressing things like eomes and hobbit and Tibet. And I'll talk, as I say, talk a little bit more about what that might mean. But the naive populations didn't have hardly any of these sort of effector-like molecules, but did express things like TCF7 and Helios. So to sort of bring all some of that together and sort of try and bring it under sort of like a hypothesis, and this is what we put forward as maybe what's happening with these V delta 1 repertoires. So in the circulation, you've got quite a mixed population of naive and clonal populations, and these naive cells might actually be driving towards lymphoid tissue using molecules like CCR7 and CD62L. and being drawn there by their chemoattractants such as CCL19 and 21. And obviously these naive cells express the IL-7 receptor, and lymphoid tissue is one of the key sites for this. So we propose that there's a naive population circulating and also entering these lymphoid tissues. And then conversely, the clonal population may well be trafficking to peripheral tissues. We've done a bit more extensive work around this, and we do find clonal populations that are in the blood and tissues, and I'll talk about that a bit later. But they were highly sensitive to IL-15, which is a key molecule in many tissues, and particularly in sort of inflammatory and infection sites. And we think these might be recognizing microbes. But again, that's a bit of a hypothesis, and I'll talk a bit more about data in the context of malaria. So that sort of brings us nicely to this, and I don't think I had a question there, but this sort of brings us nicely to our study and the work we performed within the context of malaria, and sort of looking at whether gamma delta repertoires were driven by malaria and whether there was selection and differentiation. And this was work done by a postdoc in my lab in Monash, and two Ph.D. students who've now joined me at Warwick as postdocs, Priyanka and Daniel. And so they got through an absolute mountain of samples and work over a good few years to generate this data. So I'm just going to quickly introduce malaria. I'm not going into too much detail, but needless to say, it's a major pathogen that causes a lot of infections. So these numbers probably need updating, when I think it's actually increasing for the first time in about 6 or 7 years. The number of infections are increasing. It's probably because of effects on global warming, and actually the deaths per year that are attributed to those malaria infections are also on the rise. So they've picked up quite significantly. So I think we're at about 650 to 700,000 deaths a year, and a lot of the community are hoping that's going to dip back down. But investment in anti-malarial strategies and anti-mosquito strategies have really dropped in the last few years. So there's no real end in sight there for a lot of communities and populations in Africa particularly. So as I said, there's only one licensed vaccine. So there's a major need, and the World Health Organization have mandated that we identify a vaccine that's over 50% effective immediately in the next 5 years, hopefully. But one of the key challenges with malaria is the fact that it's a multi-life cycle parasite, a multi-stage. So it lives also in a vector, so the Anopheles mosquito, particularly for Plasmodium falciparum, which causes the most deaths a year out of all the sort of malarial parasite species. And so the key to malaria as infection strategy is that it infects a human via a very small number of sporozoites that are injected into the skin. And then these travel to the liver, where it engages in a liver stage cycle, which is clinically silent. So there's no sort of symptoms or anything like this, where they enter into hepatocytes and then rapidly develop over the next 7 days before bursting out as a merozoite population, and then starting a rapid asexual stage or cycle of infection of red blood cells. And some of these cells will turn into gametocytes, which then are picked up by new mosquitoes, and the cycle continues. And so the immune system's been implicated at multiple stages, particularly the liver, which is a very key site that's been identified for blocking generating a blood stage, because this is the main stage where symptoms start, and some of the key effects of malaria and sort of that would cause death and severe pathology in individuals. And so gamma deltas have been identified to be involved in multiple stages, as well as CD8 T cells and other populations like NK cells and NKT cells. Much of this is drawn from mice, so it's not completely clear in terms of gamma deltas what exactly is happening for the in humans, and so I'm just going to talk a little bit about that. So let me just check that one. Yes. So and one of the things that really got us interested in sort of gamma deltas, and particularly sort of maybe some other populations of gamma deltas, is some prior studies. One that we were involved with, which was looking in the liver. So as I mentioned, the liver is a critical area of immune surveillance for malaria. And in this study, we found that these circulating populations of naive and effector gamma deltas actually enter the liver quite readily, and we could find the same receptor pairings of effector cells that were found in the peripheral blood, also in the liver, but we also found sort of resident populations of clonally expanded V delta 1s which weren't sort of populated. So we reasoned that actually V delta 1s might be important in liver stage immunity, potentially. And then there was this study several years ago by some of our now collaborators using an intravenous injection of sporozoites, so Plasmodium falciparum sporozoites that were attenuated. So they would get to the liver, but then get no further. And one of the key things they found in this study was that gamma delta T cell numbers correlated with protection, and it was one of the only cellular correlates of protection. So you can see here that, you know, in dose escalation parts of the study, that it was only when you actually got over a certain threshold of gamma delta expansion that you were actually protected. And it was actually in all 6 individuals in this study that were protected, which is quite remarkable, really. And so that sort of piqued our interest. And so the sort of literature around gamma deltas in immunity to malaria at the time was sort of really focused on V delta 2 T cells and this innate response. And that's quite simply because malaria parasites make a vast amount of isoprenoids, which are generated through this MEP pathway for lipid synthesis. So obviously they're dividing heavily, so they're reliant on this pathway to generate a lot of their sort of key molecules to expand and divide. And one of the intermediate molecules, HMBPP, so this is the phosphate containing molecule, it's absolutely tiny, drives these V delta 2 responses quite vigorously. So V delta 2 gamma deltas will expand rapidly in response to this in vitro, and you know, you can get some really big numbers. And they've got some key lytic functions, and so there've been a couple of really sort of critical studies, and particularly this one by Caroline Junqueira, which indicated that V delta 2s really target the blood stage of malaria, and you can use lytic molecules to effectively kill parasites inside red blood cells. So really that has really driven what we understand about gamma deltas, and that's been much of the focus. But we also reasoned, you know, from a few studies that were done quite a few years ago by Lars Havid and colleagues, that actually saw V delta 1s expand as well. And so we reasoned that is malaria also sensed by what we were terming at the time adaptive gamma delta T cells, so particularly V delta 1s. And so to sort of start this off, we looked at a cohort of individuals that from Mali. So this is an endemic area where there's high transmission of seasonal malaria, and it's well understood in the field that actually in these areas, individuals that are born into these parts of the world where you have all this transmission, that they get repetitively infected over a number of years, and actually their immunity or perceived immunity or clinical immunity increases to a point where the sort of likelihood of severe disease and symptoms is actually greatly reduced. And so a lot of the adult population are almost completely immune. So they're infected every year, multiple times, yet they have no symptoms and don't have complicated disease. So we sort of paired this with quite a crude population from that we got at the time in collaboration with our colleague, Dr. Ed Giles at Monash Health. So age and gender matched children in Melbourne, which, by and large have not had any history of exposure to malaria, or shouldn't have done anyway. But obviously they're geographically and ethnically distinct from these children and adults or adolescents in Mali. And so one of the questions we wanted to ask was, what are the gamma deltas look like in these individuals? What's the trajectory of these cells in these malaria exposed? But obviously they're exposed to a lot of other things. So one of the most striking things actually we found initially, and it sort of repeats a few observations in the field, but we sort of maybe expanded that a little bit, was that V delta 2s basically looked very similar between both cohorts. Obviously they're very different cohorts, but they looked very similar, and their trajectories were by and large identical. Whereas in the Malian cohort, we had a significant increase in the sort of frequency of V delta 1 cells that were circulating, and this can be for a number of reasons and potentially malaria. So we looked a little bit further at this, and you can see the sort of differences in these V delta 1 frequencies are very different from the sort of Australian control population. And when we started looking at some of these ideas around sort of naive and effector cells. So one of the key things about an effector V delta 1 is that they express this molecule CX3CR1. And so we looked for these effector cells, and they were significantly increased in this Malian population, even in very young children, suggesting that they were shifting very rapidly and very early on to this effector-like phenotype. And there were some differences in their V delta 2s, but we couldn't discern too many. But this is work that's ongoing at the moment. And so one of the key and critical things that we tried to do here was look at their T cell receptor repertoires. So looking at the Australians, particularly the V delta 2 negatives, we found that actually the Australian children were highly diverse and similar to what we'd seen in the past. And you can see here when you start looking at their the occupation of the total gamma delta repertoire by the first 10 clonotypes. So, if you're highly expanded, you would expect probably the first 5 to 10 clonotypes to take up quite a significant proportion of the whole repertoire, which would indicate, large clonotypes being expanded. And you can see this here in these Malian populations, whereas the Australian children were really low. And one of the key things we did notice was in these repertoires, when we looked at the V delta 2s, they were actually very much identical, basically. So we saw the same germline CDR3 sequence, this CAL-WeVQ, in all of these individuals here, and you can see them at a relatively similar frequency, and you can see that they had very similar total frequencies across the top 10 clonotypes. And when we started looking at diversity indexes, you can see that the V delta 2 negatives were very much lower on this diversity index, whereas the Australian V delta 2 negative populations were much higher. And so although we sorted and looked at these V delta 2 negatives, what we found was the majority of them were actually V delta 1 positive cells, which was quite interesting, because there are other delta chains that could be used. And so within this population, we were obviously able to look at a lot of individuals at a specific time point, but also we wanted to look at over time and see how this changed. And so this is quite a complicated map of the individuals, which obviously it's a very complicated disease and complicated situation. But the bottom line is we could look over 3 years, 3 malaria seasons, denoted by this wavy curve, and looked at different time points. And so just to sort of summarize this quite complex data, we found that the frequencies of V delta 1s fluctuated pretty much year on year when you had sort of symptomatic malaria episodes, whereas the V delta 2s, we captured a sort of time point where they increased initially and then not a lot happened, which actually correlated with some work by Maggie Feeney's group a few years ago and has been followed up really nicely. Whereas the CD8 T cells in the same individuals pretty much plateaued. We didn't really see too much of a change in those cells. And interestingly, when we looked at asymptomatic time points, where these individuals were infected but didn't have symptoms, neither the V delta 1s nor the V delta 2s showed any responses at baseline and 6 months later. So that was quite interesting. So then we looked at some of their clonotypes using iRepertoire's sequencing technology, and in the V delta 2s, there was a little change in the clonotype structure over time, whereas the V delta 2 negatives, and again, these were mainly V delta 1 clones, there was certainly some changes, but these could be attributed to a few things. But it was very telling to be after a malaria point where they were infected or had symptomatic infection. And just showing you some FACS plots here. So we stained some of these cells with our V delta 1 TCR antibodies and a pan gamma delta TCR antibody. And one of the things sometimes you find is that the population starts striping. And so you get sort of specific populations forming different sort of antigen densities of these receptors on their surface. And you can see here at this sort of 0 point, you can see only a few sort of cells in there which are in the red. And then 12 months later in the blue, they've sort of expanded a little bit more. And then as we've had more infections and more episodes and time has passed, you can see new clones starting to emerge. Whereas if you look at this one here at time point 32, you can see new stripes that have appeared that weren't there in the first place, or ones that have expanded. So when we sequence this using single cell paired TCR sequencing, you can see that certain clones were prominent initially, then they've maintained themselves, dipped down, the new ones have come up. So it's quite interesting looking at the sort of T cell receptor dynamics in these individuals, and this is something we've pursued recently. So one of the key things about these sort of populations is that obviously they're exposed to multiple other things. It's not just malaria. And so there's quite a bit of literature on cytomegalovirus that drives V delta 1 responses. So that's a major confounder in these populations. And we're not too sure if that has an impact or not. Maybe it does, maybe it doesn't. But it was very telling that you were seeing expansions post acute malaria episodes. So one of the things we did, and we're very fortunate to work with Professor Kirsten Leich and Kim Williamson in Maryland, they'd conducted a trial where they'd done a repeated controlled human malaria infection called a CHMI in a group of individuals. So essentially each individual volunteered to be infected with malaria, and they came back on 4 separate occasions to be infected, essentially. And so we got PBMC samples from these individuals, and you can see a schematic of the 5 individuals we looked at. And you can see CHMI 1 through to 4, all 5 of these individuals attended for all 4 CHMIs, which is quite remarkable. And this occurred over about 3 days, 3 years, sorry, not 3 days. That'd be incredibly short. Over 3-year time point. And you can see some of the dynamics, and you can read up on some of this in our paper. So one of the key things we found was that after CHMI 1, most of the individuals had quite severe symptoms. They had, on occasion, high fevers. But as you move through this repetitive challenge study, by the end of it, 3 out of the 5 individuals were completely asymptomatic or essentially tolerant to malaria, even though they had high levels of parasites in their bloodstream, which was quite interesting and something that sort of we took on further. And so one of the things we then did was use flow cytometry to look at the gamma delta compartments, and we found that gamma deltas increased rapidly over time and over the different infections. Essentially, gamma delta numbers went from less than 5% at baseline to nearly 20% by about CHMI3, which is quite remarkable. And one of the key things we noticed was that most of this was driven by V delta 2 responses. So there was a lot of cells, but those V delta 2s dropped off towards CHMI4, which was the key time point when people were starting to become more tolerant to the infection. And this was the point where we saw V delta 1 starting to expand in a subgroup of individuals. So there was 3 individuals where they were expanding. They became about 6% from less than 1% of these of the circulating T cells. But there was also a group that didn't really respond that well. So they were starting to, but they weren't really responding. And so we sort of grouped these out. And we found actually that the individuals that had no symptoms by the end of the CHMI4 were the ones who had V delta 1 expansions, whereas the ones who were still having symptoms, or mild symptoms at least, were still symptomatic. And these were the ones where they had no V delta 1 responses. And also there were some very interesting V delta 2 responses, whereas the ones who were asymptomatic had a very robust, consistent response that dipped at CHMI4. So the only thing that really correlated, and we looked across multiple different cellular correlates here, was this V delta 1 response. So that correlated with reduction in symptoms and tolerance, which was quite interesting. But V delta 2s didn't, CD8 populations didn't, and we looked quite extensively. So this sort of set us up that actually, maybe it's quite important to have a robust V delta 1 response. But what was that? What was those V delta 1s doing? Obviously, we'd looked at it quite significantly in the past in sort of healthy individuals. So now we have the perfect setting to start looking at that differentiation between these naive populations and effectors. And obviously, we'd established some nice markers to do this. So, for example, we can see here that in this subject 4 at baseline, the majority of the cells are expressing CD27 and CD28, which are hallmarks of this naive population. But by the time they'd ended the 4th challenge, or after the 4th challenge, almost all of those cells downregulated CD27 and 28. And as I mentioned earlier, this molecule CX3CR1, which is a homing receptor to tissues, was almost completely upregulated in this population of CD27 CX3CR1 positive cells, which directly correlates with this sort of clonal effector population we'd seen in healthy adults in prior studies. So it was quite remarkable, this differentiation, and it seemed to happen not initially very well, but it was gradually built up, and it seemed to be building with this repetitive infection. And you can also see here quite critically that their killer function, so granzyme A, granzyme B, and perforin expression were, exponentially increasing as the repetitive infections increased. But other granzymes, such as granzyme K, which its function is not well understood, didn't change. So there were certainly markers that didn't change that were sort of irrelevant to this effector response, but ones we'd seen before that did change were really important. And so it sort of suggests maybe they're engaging in anti-malarial killer functions. And so coming back to the repertoires, we sort of set up a really nice idea that these cells are changing. They were changing in the response to this controlled human infection, which is an incredibly powerful model. So then we looked at the repertoires of V delta 2 cells, and we sort of skimmed off the top 20 clones because we knew that the clones are highly shared. And you can see actually as the frequency of these cells increase, so obviously they're increasing quite dramatically from less than 2% to almost like 23 in this individual. The types of clonotypes that you can see within there don't change. They're the same ones. So it suggests polyclonal expansion of those cells, and likely to be driven by responses to this HMBPP and phosphoantigens. And this was repeated in multiple individuals to varying extents. But when we started looking at the V delta 2 negatives, and I can say this one was mainly V delta 1 when we looked at the sort of expansion, but in fact they had this large sort of V delta 8 clone that was expanded in this in the baseline, which was quite interesting. So, you know, we had a marker already that this individual had a specific clonotype we could track across time. And so what happened as they started into the CHMI, so CHMI 1, 3, and 4, I'm showing here, you can see this V delta 8 clone here, here, and here. But as you can see, it's not expanding. It's being diluted down and contracting. And in fact, what we were seeing was this sort of massive expansion almost of sort of diverse clones. So if it was a polyclonal response, you'd expect this V delta 8 clone to be equal across all of these to some extent at least, but actually there was different things happening. So we started mapping the individual clonotypes to see what was going on, and it was quite interesting because it's almost indicated that we were having waves of expansion of different clones, so sets of clones that were coming up at each individual CHMI infection. And they were actually very different. So we tried to track individual clones. Some were actually conserved. So we'd see ones emerge here at very low frequency, and they'd be they persist into the fourth CHMI. So we were seeing them again. So they were being retained. But a lot of what was coming up into the sort of the most dominant sets of the top 20 clonotypes were very different. So it's suggesting these waves of selection that was sort of and essentially drowning out this clone basically, and we saw this across multiple individuals. So in a final sort of set or flurry of experiments that we did towards the end of this study was trying to sort of piece together or loop back together, you know, what these cells are doing. So we're suggesting that they're malaria infection, particularly repetitive malaria, is driving an effector phenotype. It's driving selection within the gamma delta repertoire. But does this translate into those cells now being malaria reactive? And so to do this, we set up some in vitro experiments. So we took PBMCs from malaria naive individuals and also blood from malaria exposed individuals, so repetitively exposed. So this included CHMI donors and also individuals from Mali, where there's endemic exposure. And so we did some sort of healthy donor analysis initially, which was in bulk PBMC. So we were allowed to see V delta 1 responses and V delta 2 responses from the same individual at the same time. And so the material we used here was Plasmodium falciparum infected red blood cells, and we also developed lysates. So we took a leaf out of Maggie Feeney's book, and we generated some lysates from these red blood cells and the control lysates. So just to sort of explain the name, so the lysate would have been this PF schizont trophozoite extract. And then you'd have red blood cells uninfected, so it would be URBCs, and then you'd have PF RBCs, which would be the plasmodium ones. So if we look down here, the V delta 1 cells basically didn't proliferate in response to anything, essentially, from healthy donors. So there was no response from the V delta 1. So, that was fine, but it was all from healthy individuals. Whereas there was a really nice response by the V gamma 9 V delta 2s to the PF extract and the intact red blood cells, although this was quite variable between individuals. But there was no response to the uninfected material, so the intact or the lysates. So then we moved on to the malaria exposed individuals. And I suppose to sort of cut a lot of sort of flack spots short. So what we have here essentially is both V delta 1 and V delta 2 populations sat next to each other. We have uninfected red blood cell extract, then we have Plasmodium red blood cell extract, and then we have the intact red blood cells, so uninfected here, and the Plasmodium falciparum RBCs. And so this was in a Malian subject. And what you can see that's most striking here is that they don't really respond too much to the intact red blood cells, but the V delta 1s did start proliferating in response to the extract. And you can see that the V delta 2s were just expanding to both. And so we did this over several individuals, and we've actually done a few more of this, and it's holding true. And basically what you're finding is that the repetitively exposed individuals are responding heavily to this lysate, but they don't seem to respond too well to the red blood cells. So it suggests that there could well be some reactive populations of what definitely suggests that they're reactive. But maybe they're reacting to sort of antigens that are being presented or proteins that are being presented. And this was quite an interesting finding for our whole study as a whole. And so that sort of brings me to the end, and I'll just quickly summarize some of these things. So, it's certainly the case that V delta 2 innate like T cells are responding rapidly to malaria infection, and this is possibly driven by the quantities of HMBPP that blood stage malaria are secreting into the bloodstream. But conversely, there's certainly a shift in how the adaptive gamma delta compartment is behaving. And so we're getting shifts from this naive population. So you're getting rapid differentiation across multiple infections to these effector-like cells, which are expanding in number. And also, if you're looking in these Malian individuals, there's highly expanded clonotypes in there. So potentially they're being selected further later down the line. And they're also reactive to Plasmodium falciparum extracts and potentially some antigens there. So I suppose one of the questions that we're sort of asking now is, what does this all mean, and how does this adaptive response work, and where does it work? And, we've got some ideas that maybe it might be in the liver or different sites of the blood, potentially the spleen. And so we're looking to answer some of these questions now. So that just brings me to the end, and just to make some major thank yous to some of the many collaborators, as you can imagine, that have been involved in delivering some of this work. So particularly the past members of my lab, so Anoop did a huge amount of work, but also the current lab who are pushing a lot of this work forward, and hopefully we'll have some new exciting results coming out soon. Also my colleagues at Monash, so Jamie Ross, John Ben Gully, Jerome and Michael for some various other studies. And Ed, who's been a long-standing collaborator on local sort of cohorts in Monash, but also our international collaborators, so particularly Kirsten and Kim for the CHMI trial, Peter Crompton. We've had a very long interaction for a number of years, and his Mali cohort are absolutely fantastic, but also other people such as Joel, Cheryl for some key samples that helped us to sort of understand this, and Justin, who provided our Plasmodium falciparum infected red blood cells, and also where I did my postdocs with Ben Wilcox. We started a lot of the sort of basis for this work, and a lot of this is ongoing, and we still collaborate today. So that just leaves me to some of the funding bodies that supported some of this work, and also support the ongoing work that we're doing, and also iRepertoire for the invitation to give this webinar today. So hopefully you all enjoyed it and learned a bit about gamma deltas, and particularly their response in malaria. And if anyone would like to explore this further, or is interested, then here's my email. I'm easily findable on Google if you stick in Martin and Davey and Warwick. But yeah, if you want to get in touch, then here's my email address. And that just leaves me to thank you for your attention. And if you've got any questions, I think there's the Q&A going on. So thanks very much.
Thank you, Martin, for a really great presentation and also a pretty interesting one. And as such, we have some questions that the public had just asked. Just as a reminder, please submit your question using the Q&A as Martin is answering the first few questions. So the first question, Martin, that we have is, do you think there is more merit to look at the Delta repertoire instead of the Gamma repertoire?
Yeah, I suppose, it's certainly, if you're looking at sort of V delta 2 repertoires, I think looking at the Delta repertoire is quite informative, because we have actually seen changes in the Delta repertoire. So the Gamma repertoire is quite conserved. But I suppose looking at the Delta repertoire, for example, in V delta 1s, I actually think you should be looking at, both at the same time, because there is some distinct changes. And if you can do some paired analysis as well, that's also useful, because it really does, because there is quite overt clonotypes that are being picked up. So picking up the chain pairings and what's there is actually, I think, really important, particularly for sort of antigen discovery potentially and other sort of responses. So I think actually doing both is the way forward.
Perfect. We have another question that says, can the PCR data be used to predict the antigens that they are reacting to?
Yeah, that I get that question a lot. So there's not a lot of firstly, the data that's out there for sort of alpha beta T cells, and you can use repertoire data to attempt to predict antigens, and a lot of that's been based on the structural studies that identify the peptides and the MHCs, and so identify lots of repertoires that would do that. Whereas there just isn't really that data in sort of the gamma delta field, and there's definitely not the depth of understanding of the antigens. So to predict an antigen, I think, is incredibly hard. Maybe we can take some sort of some ideas from the B cell field more realistically, but even then I think antigen prediction is quite difficult because of the you get degenerate recognition, so basically recognition of multiple epitopes on the same protein, for example, which then means that what is your T cell receptor, for example, if they're doing that, what are they seeing and where? I think it's quite hard to do. So to answer your question, no, you can't at this stage, but it would be amazing if you could.
Yeah, absolutely. So we have a question that actually starts with amazing work. Since the PD-1 cells express a lot of cytotoxic marker, is there any correlation with their expansion and the percent of parasitemia?
Yeah, so in our CHMI study, that's probably the best one to look at for that kind of thing, because obviously we the clinical site measured the levels of parasitemia in those individuals, and despite the massive increase in sort of lytic or cytolytic gamma deltas, and particularly V delta 2s, there was no change in the parasite load in each of those individuals. Their symptomology changed, but their parasite numbers didn't. So I think that's quite interesting in itself.
Yeah. Absolutely. And another question that we have is, what do you think is the contribution of bystander activation on all of these responses?
Yeah, that's a good question. Yeah, I suppose these are quite inflammatory scenarios, and there's a lot of cytokines and things. But we've been asked that question a lot, particularly by reviewers and in the past, and we've tried to look at this in sort of effector V delta 1 responses, and they don't seem to be the differentiation doesn't seem to be driving or driven by cytokines. So you can't drive a naive cell with a whole heap of cytokines towards this effector population. You can drive those naives into some weird states where they still retain CD27 and various other things, and a few other groups have done this in other settings. But it's not entirely clear. So the V delta 2s, I think that's more clear cut because they're responding to they can respond to IL-12, IL-18 and things like this, more innate cytokines, so similar to NK cells, etc. But the V delta 1s are less responsive, and in fact, when we've looked, they don't express half of those receptors, so there's no chance of them activating to IL-12 or IL-18 because they don't have the key receptors for those cytokine responses.
Perfect. Going along similar lines, have you compared any of your TCR to non-CMV reactive sequences?
Well, if you can show me what a known CMV reactive sequence is, then maybe we would. We've looked at them in so there was one a number of years ago by David Mylan's group in Belgium. So this was a in utero receptor that was or congenital CMV receptor, and it was he quite clearly demonstrated it was reactive, but we've never found it expanded in any of our cohorts. And there was another paper in Nature Immunology by Imo Prince's group, and they showed CMV drove some clonotypes in a stem cell setting, or stem cell hematopoietic stem cell transplant setting anyway. And we haven't seen any of those clonotypes. And even when you look in between individuals, they're almost completely private in terms of what you see in receptor usage, which is quite interesting as well. So no is pretty much the answer to that.
Perfect. So thank you very much, Martin, for your great presentation again and for answering all those questions. That's all the time we have for questions so far. So if we didn't get any of your questions, we will reach out to you through email. And if you have any additional questions about Martin, you can please write to him or Google him, as he mentioned, or you can please write to iRepertoire with the information that we are showing on the screen. And please stay tuned for more webinars that will come. And again, thank you very much, Martin. And we thank you, all of you, for attending today's webinar. Goodbye and have a great day.